Good day, everyone. Today, we're going to discuss another topic in STS, which is on gene therapy. So, it's very controversial. It poses a lot of ethical questions in here about regarding gene therapy, but it also brings a lot of potential benefits. So, today we're going to explore what are these benefits at the same time, what are these risks. Okay, so specifically, this is our intended learning outcome. You will be able to evaluate gene therapy, its various forms, issues, potential benefits, and detriments to global health. Okay, so in 1991, James Watson declared that many people said that they were worried about the changes in their genetic instructions, but these are merely a product merely a product of evolution, shaped so they can adapt to certain conditions which might no longer exist. Okay, when we hear the word evolution, we normally associate this one with macroevolution, meaning to say those which are tangible. But even even microcellular structures or such as our DNA. So these DNAs are also evolving in a way that we need to adapt to certain conditions of our environment. Okay, so let's take this example. For example, if before climates or weather were too cold, and as time goes by, temperature increases, and so our body needs to adapt with this climatic condition. And again, this has to be predetermined by our genetic composition. Otherwise, if there is no evolution with respect to our genetic compositions, well, we could not live this far. In other words, we'll die. Okay, so we all know how imperfect we are. That's why our genes have to evolve. So why not become a little better apt to survive? Okay, so um, at least if we might not be perfect, but at least we can survive. Okay, and again, that's the importance of evolution. Okay, so since the beginning, humans understand that the peculiar characteristics of the parents can be transmitted to their descendants. The first speculation originated from the ancient Greek students, and some of these theories continued for many centuries. Okay, so this is one good example, which proves that um, uh, characteristics can be inherited or transferred from one generation to another. So in 1850, when an Austrian monk named Gregor Mendel conducted a series of experiments with green peas. Okay, so basically, what he did was to cross pollinate um, two pea plants of different colors. Okay, so one is yellow and the other one is green, and it is thought that yellow is dominant over green. So during cross pollination and the product of it in the first filial generation, so all offsprings. Again, after cross pollination, expressed yellow in color. It's because of the dominance of the yellow color. However, when these first filial generation self-pollinates, okay, so we see here three allelic combinations. One is homozygous yellow, meaning to say it's pure yellow. The other one, that's 25%. 50% is heterozygous yellow, which means the alleles or genes for green is present but it is not expressed. It's because again yellow is a dominant color. Okay, and 25% chance that the offspring will become green. Okay, so again by crossing the two heterozygous yellow during the first filial generation. So what I'm trying to say here is that this is a good example that again traits can be inherited and it can be transferred from one generation to another. Because if in the event that there was no transfer of these attributes or these characteristics contained within the DNA, well, most likely um, we could expect different results. So instead of yellow or green, it could have been red or blue. Okay, so up until 1950s or 1950, little was known as to the physical nature of genes, which was when the American biochemist James Watson and British biophysicist named Francis Crick developed the revolutionary model of a double-strand DNA. So in 1970, researchers discovered a series of enzymes that enabled the separation of the genes in predetermined sites along the DNA molecule and the reinsertion in a reproducible manner. These genetic advances prepared the scenario for the emergence of genetic engineering with the production of new drugs and antibodies, and as of 1980, gene therapy has been incorporated by scientists. Okay, so gene therapy then is an experimental technique that uses genes to treat or prevent diseases. So in future, this technique may allow doctors to treat disorder by inserting a gene into a patient's cells instead of using drugs or surgery. Okay, so there are three basic approaches as to how gene therapy is conducted. One is by replacement of a mutated gene that causes a disease with a healthy copy of the gene. Okay, and the other one is to inactivate or knock out a mutated gene that is functioning improperly. And then the third one is to introduce a new gene into the body to help fight disease. Okay, so although gene therapy is promising treatment option for a number of diseases, including those inherited disorders, some types of cancer, and a certain viral infections, the technique remains risky and is still under study to make sure that it will be safe and effective. Gene therapy is currently being tested only for diseases that so far have no cures. Okay, so in other words, only selected diseases. Okay, so um, uh, not all diseases are currently being studied as to the effectiveness of these gene therapy. Okay, so but again, it is still risky, although there are preliminary studies as to its side effect, but we don't know its long-term effect. Okay, so so far these are the diseases. Table one shows gene therapy protocols, and it also shows some diseases which so far are are being applied with gene therapy. Okay, so we have adenosine deaminase deficiency, alpha 1 antitrypsin deficiency, AIDS, different types of cancer, cystic fibrosis, familial hypercholesterolemia, Fanconi anemia, we have Gaucher disease, hemophilia B, and rheumatoid arthritis. Okay, so these are the respective objectives of these diseases, and these are the sources of stem cells. Okay, so and so far, as you can see here, the countries with the protocol on how to carry out gene therapy with these diseases are mostly developed countries, are all developed countries rather. So such as Italy, Holland, United States, you have Austria, France, China, Germany, Italy, and among others. Okay, so so far, there's no there is no Southeast Asian country yet. Okay, so the question now is, how does gene therapy work? Okay, so let's take a look at this short video presentation.
Genes make us who we are. Are we tall or short? Do we have blue eyes or brown? Our genes hold the answer. What if part of a gene is defective or missing? Or if a gene changes or mutates? It can cause a disease like cancer. Enter gene therapy. Among other things, scientists can replace a bad gene with one that is healthy, add a new gene to perform the function of a defective or missing gene, or they can turn off a problem gene. How do they do it? Say scientists want to add back a healthy copy of a gene to cells. They need an efficient way to do it. So scientists use vectors or vehicles to deliver a gene. The healthy gene hitches a ride in the vector. Viruses are one kind of vector. Scientists modify a virus to remove its ability to cause a disease. The modified virus can then be used to carry the healthy gene into a cell. Gene therapy can happen on cells inside the body, or can happen on cells outside, and then transplanted back in again. Genes make us who we are, and gene therapy holds great promise for a healthy future.
Okay, so you heard the three approaches of gene therapy, such as replacement, knockout or inactivation, and of course the introduction of new genes. Okay, so for gene therapy has to be, or for the gene for genes to be introduced into a cell, it needs to have a vector, so such as a virus. Okay, so
Brown. Our
okay, so now when we look at that, or if we are to convert that video into a diagram, so this is how it looks. So as you can see, this is a virus. Okay, so a virus which gene has to be introduced. But of course, these viruses has to be modified in a way that it could no longer cause diseases. Otherwise, if we're going um, if we're not going to modify the virus, well, it will just cause certain disease. So that's the preliminary thing which scientists have to work on. So to modify the virus in a way that it will not cause diseases to us humans. Okay, so what happens next? So this virus becomes a vector, a vector to transport the genes. Okay, so these virus rather would eventually um, would eventually penetrate into inside the plasma membrane until it reaches the nuclear pores. Okay, so in the nuclear pore, the the virus disassembles and delivers DNA into the nucleus. Okay, so as it disintegrates again, the DNA is now inserted inside the nucleus. Okay, so genetic information from the new gene is transferred to a molecule called mRNA. So this serves as a blueprint for protein production. Okay, so these are some of the viral vectors for gene therapy. We have retrovirus, lentivirus, herpes virus, adenovirus, adeno-associated, and plasmid. Okay, so these are the parameters which they can be compared of, such as provirus, their capacity, integration into the recipient genome up until the germline transmission. Okay, so by the way, virus is only one form of mechanism or approaches for genetic modification of the cells. So again, virus mediated is one, but we also have the physical mechanisms. Okay, so the question that we have now in our mind is gene therapy safe? Well, gene therapy is understudy to determine whether it could be used to treat diseases. Current research is evaluating the safety of gene therapy, and future studies will test whether it is an effective treatment or treatment option. So, several studies have already shown that this approach can have a very serious health risk. So, these include toxicity, inflammation, and cancer. So, um, even though it is used to treat cancer, but chances that it might it might lead to the development of another cancer, or worse, it might uh, what how do we say that it might cause the cancer cells to become to mutate more. Okay, in other words, it just worsens the situation. So because the techniques are relatively new, some of the risk may be unpredictable. It's really true because it is not just like a chili which if we if we're going to eat that one eventually we feel that it's very spicy, so it takes time. So with that, some of the risk may be unpredictable. However, medical researchers, institutions, and regulatory agencies are working to ensure that gene therapy research is as safe as possible. Okay, so with that, there are comprehensive federal laws or regulations and guidelines to help protect people who participate in research studies, or we call it as clinical trials. Okay, so for example, we have the US Food and Drug Administration. So they are the ones responsible in regulating all gene therapy products in the United States and overseas research in this area. So researchers who wish to test an approach in a clinical trial must first obtain permission from the FDA. So the FDA has the authority to reject or suspend clinical trials that are suspected of being unsafe for participants. Okay, so again, that's the reason why we have as I present the one which I presented a while ago, which you can see some protocols. These are actually protocols as to how to carry out studies about gene therapy. Because if in the event that the certain process would actually um, put put the participants' health at risk. Although chances, because in the first place, there's really a chance that there that the particular intervention would really worsen or would cause a negative impact to the health of those participants in the clinical trial. But at least the risk is being minimized. Okay, so um, these institutions, aside from FDA, we also have the National Institutes of Health, which also plays an important role in ensuring safety of gene therapy research. So it provides guidelines for investigators and institutions, um, such as universities and hospitals. Okay, because uh, research does not just happen inside hospitals for gene therapy, but it might as well it might be conducted also in some universities. So there should be they should follow when conducting clinical trial. So there should be some guidelines which these researchers have to follow when conducting clinical trials with gene therapy. So these guidelines state that clinical trials at institutions receiving funding from NIH for this type of research must be registered with the Office of Biotechnology Activities. So the protocol or plan for each clinical trial is then reviewed by the NIH Recombinant DNA Advisory Committee to determine whether it raises medical, ethical, or safety issues that warrant a further discussion at one of the RSA's public meeting. Okay, so we also have the Institutional Review Board. So we have FDA, we have NIH, and now we have IRB. So, and also Institutional Biosafety Committee. So they must approve each gene therapy clinical trials before it can be carried out. So an IRB is a committee of scientific and medical advisors and consumers that reviews all research within an instruction. An IBC is a group that reviews and approves institutions' potentially hazardous research studies. So there will be multiple levels of evaluation and oversight ensure that safety concerns are a top priority in planning and carrying out gene therapy research. So these IRB, in fact, we do not just limit with experts with the topic and some medical advisors or scientific experts. We even extend to other professions, so such as those in the law professions, um, and some even tap with religious sectors. It's because for science, the guidelines or the protocols may be very sound, but with respect to those lawyers, well, they will also look into other aspects of the research, such as its legality. If that particular clinical trial would not violate human rights, for example. And for the religious domains, so they are concerned as to its ethicality, its morality. It's because um, with uh, with uh, with the case of the lawyers, the protocol may be sound, may be legal, but not all legal are considered as moral or religiously sound. So these are the things that we need to weigh out before they come up or before they give their verdict that such research study can be conducted, or it will proceed, or yes, it will proceed. Okay, so what are the ethical issues surrounding gene therapy? Okay, so one is, how can good and bad use of gene therapy be distinguished? Okay, so who decides which traits are normal and which constitute a disability or disorder? Okay, so um, for me, as a postmodernist, I really consider myself as a postmodernist. It's a school of thought in which sort of a very, very skeptic. For example, there's an idea, usually postmodernists says, so who validates that idea? So and by that, in other words, we become skeptic. But anyway, it's a school of thought. Okay, so much like this one. Okay, so um, we cannot say that that's a particular, for example, that a certain trait is normal, or or it is it's a disability, or it's a disorder. Okay, because let's take this case, being beautiful. Okay, so being beautiful, of course it's normal. um I mean, being let's take being ugly. Okay, so although ugly is a subjective term, because we have definitions, or we have different descriptions what really ugly is. So for some, um, it is not considered as normal, and it may be considered as a disability. Okay, but for others as well, well, it's just normal, and they do not consider it as a disability. Okay, so much like gene therapy, it may be, for some it may be normal, but for some it may be not. Okay, so that's one ethical question. Second is, will the high cost of gene therapy make it available only for the wealthy? Well, it's certainly true, because with the social structure that we have, in which there is a large gap between the rich and the poor. So certainly, if ever these will become available in the market, so those who will take advantage of this are, of course, the rich people. Because for us, living in the in the marginalized sector, our primary concern is, of course, to satisfy our basic needs, and we cannot afford this one. Okay, so again, this is also one ethical question that we have to consider. Next is, could the widespread use of gene therapy make society less accepting of people who are different? It's because if gene therapy becomes legalized, and so um, uh, we will genetically engineer ourselves in a way that we will become perfect per se. Okay, and those who cannot afford, well, they would become different. They would not fit into the norm, and so there will be discrimination, as they might not be accepted. Next is, should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? Because if this is the case, that all should be genetically engineered, then there's no point of having Olympic Games, of having quiz bee, and among other competitions which involved our intellect, or our athletic ability, and even our height. Okay, so there's no need to have a basketball if all players are, let's take, 6 feet, measuring 6 feet. So basically, they can just they are more or less, they're of the same height with the height of the rings. Okay, so again, these are some things that we need to consider, or these are some ethical questions which we need to consider, or which we need to consider with gene therapy. Okay, so um, another controversial or ethical issue, really, which is associated with gene therapy, is this germline therapy. So, with this type of gene therapy, the target are the egg and sperm cells. So, which allow the inserted gene to be passed to future generations. So, when we look at the diagram, this is how it looks. So, what makes it controversial is that while it could spare future generations in a family from having a particular genetic disorder, because let's just take it, that that a certain disorder runs through your genes, and so this has to be transferred from one generation to another. But with germline gene therapy, the good thing here is it can actually spare, or what do you call it? Yes, spare future generations to be sick with the same disease with the parental generations. But this might affect the development of a fetus in unexpected ways or have long-term side effects that are not yet known. Again, as I said a while ago, um, these gene therapy, the impact of gene therapy, although there are side effects uh, which eventually we can have, but that would eventually be felt. But then again, there are some how do you call this one? There are impacts which may prevail, or which may exist in the long run. Okay, so because people who would be affected by germline gene therapy are not yet born. So in other words, if ever there will be side effect, so they don't have any option, they don't have any choice but to suffer the consequences. And again, they were still fetus, so they were not able to decide whether they approve such germline gene therapy. Okay, so because of these ethical concerns, the US government doesn't allow federal funds to be used for research on germline gene therapy in people. However, in 2015, Chinese researchers went beyond the moral issues and announced for the first time the genetic modification of embryonic cells using CRISPR-Cas9 technique. And then another Chinese group also reported the conduction of the same process done with the intention of conferring resistance to HIV by insertion of the CCR5 gene mutation. So the genetic analysis showed that 4 of the 26 embryo were successfully modified, and the results clearly reveals that the need for improving the technique, alerting that possibly such trials could be previously tested in animal models. So with this, recent publications rekindle the debate regarding gene editing. Okay, so this is what gene editing with CRISPR-Cas9 is. So let's watch.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy. But recently, a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location. by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Okay, so um, that ends my presentation on gene therapy. So again, these are the sources which I have, and the images were also taken from various websites, and I already provided the links below each below each image rather. Okay, so your task for this topic, you're going to write your personal opinion about gene therapy in a form of essay. So you will be graded according to the following criteria: ideas, organization. By the way, ideas account 30 points, 5 points for organization, 5 points for sentence fluency, 5 points for conversion and presentation, 5 points. So please limit the number of words you contain in your essay to 300 words if possible. But if you exceed, that's fine. But again, please limit it to 300 words if possible. So all of your outputs will be subjected to plagiarism tests. I have Grammarly in here, and I will be guided if your works are copied or original. Okay, so that's all for now, and have a good day. Goodbye.